Hello, my name is Jacob Lang, and this is my submission for the National Academy Medical School Scholarship Challenge, called Treatment for Cancer Using New Antibody-Drug Conjugates. Cancer is the second deadliest disease in the U.S., and about 8 million people around the world are estimated to die from it each year. Throughout the last century, there has been an ongoing fight against this disease, and I believe a variation on current methods that I have theorized will greatly improve the number of lives saved from cancer. A form of cancer treatment using antibody-drug conjugates, or ADCs, has been showing auspicious results in the past few decades. This treatment is implemented by using antibody that binds to receptors on a tumor cell's surface and chemically linking it to a cytotoxic or drug that is capable of killing cells. It somewhat resembles a heat-seeking missile in the way it functions. The antibody acts as a heat-seeking part, while the cytotoxin acts as the explosive payload. The antibody targets the cancer cell, while the cell-killing reagent terminates the tumor cell. The benefit of this method, instead of traditional chemotherapy, is that it only kills the tumor cells and leaves the healthy cells around them relatively unscathed. And this is in great contrast to traditional chemotherapy or radiation therapy, which cause damage to healthy cells in the process of killing the cancer cells. So, the side effects of antibody-drug conjugates are also much more minor than other popular cancer treatments. However, there are a few problems with this method. For one, sometimes the cytotoxic compound is not potent enough and the cancer cells are not killed. The other major setback would be the buildup of dead cell tissue. As the cancer cells are killed, there will eventually be so much dead tissue that the antibody-drug conjugate would not be able to get to the still living cancer cells. This would allow them to continue growing and spreading, which would render the entire treatment useless. In spite of those problems, ADC treatments are already being employed today, and the results are extremely positive. One of the most encouraging examples is TDM1, or trastuzumab emtansine. This antibody-drug conjugate, which is mainly used for late-stage breast cancer patients, has been shown to greatly improve patients' health. According to the FDA, in clinical trials, results showed that patients treated with Kadcyla had a median progression-free survival of 9.6 months compared to 6.4 months in patients treated with lopidumab plus capecitabine. The median overall survival was 30.9 months in the Kadcyla group and 25.1 months in the lopidumab plus capecitabine group. This is just one example of the ADC treatment's efficacy. My hypothesis is that by using an antibody-drug conjugate made from a monoclonal antibody that binds to cancer cell exclusive receptors and the cytotoxic compound perforin in coincidence with the phagocytic signaling molecule fractalkine, the success rate in using ADC treatment will greatly increase. The first step is to determine what type of cancer it is. For an example, I'll use colon cancer. Cells from the patient's tumor must be taken in order to make monoclonal antibodies from them. Then I will bond the antibody to the cytotoxic compound perforin. Next, I will take the antibody-drug conjugate and inject it into the site of the tumor. Then, once it is attached to the tumor cell must ingest the conjugate molecule. The perforin linked to the antibody will activate, and the cancer cell will be killed. The most important aspect of my idea, and what makes it surpass other antibody-drug conjugate protocols, is the use of perforin. This cytotoxic compound has many benefits over other choices. For one, it is already being used in all of our bodies. The T cells in us use perforin to kill infected and cancerous cells. So, this solves the problem of it not being potent enough. Because it is a biological compound, instead of an artificial toxin, perforin would have a much lower impact on the health of a patient than other compounds. Another benefit is that it can cause tumor cells to undergo apoptosis, which is a type of cell death that causes no damage to the cells around it. When a cell undergoes apoptosis, it also releases phagocytic signaling molecules, which have a very beneficial function that contribute to solving the problem of too much dead cell tissue. So, perforin would be the perfect cytotoxic compound. The phagocytic signaling molecules that were previously mentioned have a huge role to play in this treatment process. As stated in the beginning, a common problem with antibody-drug conjugate treatment is the buildup of dead cells, which impede the conjugate's task of binding to cancer cells. However, the answer to this problem is phagocytes. These cells are capable of entirely enveloping other cells and digesting them, and the great aspect of them is we already have billions of them in our bodies. While they do reside in us, they can be relatively slow to react. So, at the site of the cancer, I will inject fractalkine as well. Fractalkine is a molecule that signals phagocytes to go toward wherever it is. This, used in conjunction with the other phagocytic molecules that are released from the cells going through apoptosis, should greatly proliferate the amount of phagocytes at the site of the cancer. Once they arrive, the phagocytes will start ingesting the dead cells, and since there will be so many, the dead tissue should be rapidly cleared away, allowing the ADC to continue targeting and killing the cancer cells. This progress should be repeated. Inject the antibody-drug conjugate into the site of the cancer, inject the fractalkine, then wait for a period of time and observe the patient. If no improvement is seen at first, give another dosage and wait for the results. The extraordinary aspect of this treatment is that with slight variations, it can be applied to all types of cancers. All that would need to be done is to make a monoclonal antibody from the tumor cells of that particular patient's cancer, combine it with the perforin to make an antibody-drug conjugate, and follow the previous steps. Theoretically, it would have the same desired results. The speaker that inspired me most was definitely Jack Andraka. The fact that he was basically my age, but managed to invent something as helpful as a new revolutionary test for pancreatic cancer is now continuing to be a contributing member of the scientific community really encouraged me. It made me feel as though I could make an impact like he has, and hopefully I could help as many people as he has too. This concludes my submission of the 2015 National Academy Medical School Scholarship Challenge. Thank you very much.